2019, and he was working with Dr. Andrew Delameter at the City University of New York. His research explores the computational principles behind reward-based learning and decision-making, including drug-seeking behavior and how the brain implements those computations. Informed by his background in learning theory, he aimed to review how reinforcement learning models map onto a basal ganglia function to support behaviors that span different degrees of flexibility. He's also a proud first-generation college student, and he's currently on a faculty job market. And Eric, please go ahead and share your slide.
Sure, thank you. Cool. All right. Thanks for having me. Very excited to tell you about my postdoctoral work. So the title of my talk is Learning Cause and Effect with the Help of Mesostriatal Dopamine. So this is an old picture of my son, Jude, when he was a toddler. And back then, he really liked to eat Cheerios. At the time, he was going through a phase where he was frequently getting angry and throwing tantrums. When he threw a tantrum, it was often best to distract him with something he liked, such as Cheerios. However, one of his parents, and I won't say which one, was worried that following a tantrum with Cheerios could reinforce the tantrum and cause him to repeat such behavior if it was rewarded. So to figure out whether Jude's bad behavior is being positively reinforced, we can approach this from two different points of view. From one point of view, which I'll call value learning, we just need to calculate the reward rate for tantruming. How many Cheerios can he expect in the future on average from tantruming? So I've estimated that quantity here based on real statistics. This is a positive number, so the value associated with tantruming is therefore positive and the behavior will be reinforced. But there's another point of view, which I'll call contingency learning. So to figure out the contingency between tantruming and Cheerios, we need to also consider a different conditional probability, the probability of a Cheerios snack being preceded by a tantrum. Let's just pretend that that quantity is 1. So the only time he ever gets Cheerios is when he throws a tantrum. In that case, the probability of getting Cheerios in the absence of a tantrum is 0. And we can plot the relevant statistics on a contingency table. So to calculate the contingency, we can just take the probability of getting Cheerios during a tantrum and subtracting the probability of getting Cheerios in the absence of a tantrum. That's a positive number in this example. So here, the two competing learning theories, value and contingency, they make equal predictions. But in this example, there's this unrealistic assumption. That is, whenever Jude gets Cheerios, it's always preceded by a tantrum. In reality, he gets Cheerios much more frequently in other contexts, so this number is less than 1. We can update this number down here to something more realistic, and now the contingency goes to 0. So now the value and contingency points of view make different predictions about his behavior. So if contingency is what actually drives learning, we should find that under the circumstances specified here, Jude's behavior is not positively reinforced. The jury is still out. I don't know if what actually happened with his behavior, but my theory is that it was not being reinforced. Okay, so of course I wouldn't be discussing contingency if it weren't important for associative learning. And in fact, we've known this for decades, going all the way back to the 60s. But not many neuroscientists are aware of this, and that's not necessarily their fault, because often contingency is confounded with value in our experimental setups. Take, for example, one of the leading theories of how reinforcement learning is implemented in the brain. This theory says that dopamine, which is a neurotransmitter that binds to receptors in the striatum, acts like a teaching signal to modify synapses in the striatum so that animals can gradually learn to adapt their actions to earn more rewards. And the way this is thought to happen completely ignores contingency, at least overtly. So to explain what I mean by this, let's focus on this figure from a paper in which researchers conducted an experiment in mouse brain slices. They recorded from striatal neurons in green, while at the same time activating presynaptic glutamatergic terminals in red and activating dopamine terminals in blue. You've heard the phrase that neurons that fire together wire together, and that's true in the striatum, but in addition, you also need dopamine to be released. What these researchers wanted to figure out was if they simultaneously activated the presynaptic glutamatergic neurons in red and the striatal neurons in green, how long afterward does dopamine need to arrive to induce plasticity? What they showed was that plasticity was potentiated maximally when dopamine followed pre- and postsynaptic firing at about 0.6 seconds and then gradually tapered off. as dopamine release became more temporally distant. So what this implies is that for learning to occur at the level of the synapse, there must be sufficiently tight contiguity between one set of events, that is pre and postsynaptic firing, and dopamine release. But notice how this experiment does not address whether dopamine release must be contingent on pre and postsynaptic firing to enhance plasticity. It only says that a certain degree of contiguity is sufficient to induce plasticity. And this contiguity-based account of learning is also embodied in the dominant computational models of associative learning. So under this model, notice temporal difference learning. When a stimulus such as a tone predicts future reward, an agent can learn over repeated trials to attribute value to that tone, such that at tone onset, there's an increase in value that accelerates as the expected time of reward approaches. And here, value is just defined as the expected discounted future reward. So this captures the basic idea articulated in the slice physiology paper from the previous slide, which is that temporal contiguity between events determines the strength of learning. The further away in time this tone is placed from this reward, the longer it will take to build up this value function. Now, what does this have to do with dopamine? Well, it turns out that there's a lot of evidence that dopamine computes the derivative of this function, which is called the temporal difference prediction error, or just prediction error for short. And this is just a small sample of papers that support this hypothesis. So in the work I'm going to tell you about today, I wanted to know whether dopamine dynamics are sensitive to the contingency between events, as this could possibly change how we think about learning in the brain. As I mentioned before, value and contingency are frequently confounded, so we need a behavioral procedure for manipulating contingency independent of value. I used a Pavlovian conditioning protocol with rats called outcome-selective contingency degradation. During the acquisition phase of this task, rats are exposed to two types of trials at random times and in random order. So during a trial, an auditory cue, either a pure tone or a white noise, will play for 20 seconds and terminate with 0.5 probability of reward. And each cue is associated with a distinct reward. In this example here, the tone is paired with sucrose water and the noise is paired with pellet. So the rats experience 8 sessions of these pairings and then they move on to the next phase. So during contingency degradation, the only thing that changes is that now one of the rewards is presented randomly during the intertrial interval. This degrades the contingency for one cue reward pair while leaving the other contingency intact. So in this example, the tone is referred to as the degraded cue, while the noise is referred to as the non-degraded cue. However, the values of the cues remain unchanged. Remember that value is defined as the expected future reward, and during contingency degradation, the tone is still followed by sucrose with the same probability at the same interval. What this means is that if value drives learning, we should predict that this task manipulation has no effect on behavior or dopamine dynamics. Yet when we look at the contingency between sucrose and tone during acquisition, the contingency is positive, but during degradation, it goes to 0. So I ran this behavioral experiment in rats, and what I'm plotting here is the rate of port entries during the 20-second cue period as a function of session number. The port entries made during the cue period are anticipatory responses because they occur prior to the time of trial outcome. And this is our measure of the condition response. So they learn. That's what this plot is showing. And this is from the acquisition phase, and each trial type is shown separately in different colors. In orange, we have the cue that will undergo contingency degradation in the next phase. And in blue, we have the non-degraded cue that will serve as the control. So across the 8 sessions of contingency degradation, there was a clear attenuation of the condition response during the degraded versus non-degraded cue. And just to ensure that this was an effect on learning, not just performance, I ran one reacquisition session in which the original contingencies were reintroduced and the behavioral effect carried over. So what this is telling us is that already we know from behavior alone that value does not seem to be sufficient to drive learning, and learning seems to depend on contingency. Before I get into the neural recordings, I just wanted to mention that this important point. You may be asking yourself whether this behavioral effect can be attributed to satiety, because keep in mind that during contingency degradation, one of the rewards is being delivered with an overall higher frequency than the other. In this example, the liquid sucrose is delivered randomly during the ITI, while the grain pellet is delivered only at the end of a noise trial, which means that sucrose is consumed more than pellets and may induce selective satiety, and maybe that's what's accounting for the effect on condition-poor entries. To test if that's true, I placed a set of rats through the contingency degradation procedure for one session, and then immediately placed them in their home cage where they could freely consume both grain pellets and liquid sucrose. And then during a control session, they were given the same consumption test, but it was not preceded by a behavioral session. And the idea here is that if the contingency degradation procedure induces selective satiety, then what we should find is that in this example, the rat consumes less sucrose after contingency degradation compared to the control session. And importantly, that the reduction in consumption is greater than that of pellets. What I'm plotting here is consumption as a function of reward type, where contingent indicates the reward that's completely contingent on the auditory cue. So in this example, the pellet, while non-contingent indicates the reward that's not contingent on the cue. In this example, the sucrose. Black is the control test, and white is after a session of contingency degradation. And what this plot shows is that the reduction in satiety was equal between the reward types. This means that the procedure did induce satiety, but it was of a general form, not specific to one reward. So specific satiety does not seem to be able to explain the behavioral results. Okay, so now we come to dopamine. The mesostriatal dopamine pathway is in part composed of this projection here. Dopamine neurons originating in the ventral tegmental area release neurotransmitter onto receptors in neurons in the nucleus accumbens core of the ventral striatum. To investigate whether dopamine dynamics are altered during contingency degradation, I used a procedure called fiber photometry. This requires expressing a sensor, for example, GCamp in cells, and then implanting an optic fiber above the infected cells. I first used fiber photometry to record calcium activity in VTA dopamine neurons using GCamp. And then in a separate cohort of rats, I recorded dopamine neurotransmitter release in the accumbens core using a sensor called D-light. All right, this image is showing expression of GCamp in the VTA overlapped with the expression of tyrosine hydroxylase, which is a proxy for dopamine positive neurons. The overlap is quite specific, and that's because I used THC rats for this experiment. So I'll now show you some photometry data, and keep in mind that if dopamine neurons encode a prediction error, they're at least under the simple TDRL model, there should be no effective contingency degradation on dopamine dynamics because the prediction error is a derivative of value, and value is objectively unchanged during this task. So here I'm showing the z-score GCamp signal aligned to Q onset separately for the non-degraded and degraded cues. For the majority of rats, there's greater dopamine response to non-degraded versus degraded cues. I also align the GCamp response to the reward that sometimes comes at the end of a trial. And we see a larger dopamine response for reward following the non-degraded trials versus rewards following degraded trials. And then we can also look at reward omission because recall that reward probability is always set at 0.5. So we have some trials that result in rewards, some that result in omission. And when we look at the portion of the response that's negative, that is the portion that goes below baseline, which is normally thought of as computing a negative reward prediction error. We don't see any difference between trial types, but that could possibly be due to these baseline differences between the two Q types. Right, so I did the same thing, like I said, but looking at D-light in the accumbens core, same experiment, and I replicate what I saw before in the VTA. So larger dopamine release in response to the non-degraded cue, larger response to reward following the non-degraded cue. And this time we do see a small but significant effective reward omission where now the dopamine response is more negative at the time of omission following a non-degraded trial type. So are dopamine dynamics sensitive to contingency? The answer seems to be yes. So, of course, we'd like to know, are these changes in dopamine dynamics related to behavior? What I'm showing here is a sample trace from VTA dopamine neurons relative to cue onset. Recall that the auditory cues last for 20 seconds. So here I'm showing the full duration of the cue. And notice how there are two components to the cue-evoked dopamine response, a transient phasic component and a prolonged tonic component. I used a statistical threshold to determine when the phasic component ends and the tonic component begins. And then I regressed each component onto a measure of condition responding. What I'm showing here is across individual rats in the two different experiments. It's port entry rate, the difference in the port entry rate between non-degraded versus degraded trials. So this is sort of like the behavioral effect of contingency degradation. And then the difference in the phasic signals. So the difference between cue-evoked dopamine responses between non-degraded and degraded trials. So you can think of the y-axis is representing the behavioral sensitivity to contingency degradation, and the x-axis is the neural sensitivity to contingency degradation. These two measures were positively correlated, both for the phasic and the tonic components. As an aside, these measures are averaged across the entire session. When we actually look to see, when I look to probe whether there's any evidence of a trial by trial relationship between behavior and dopamine dynamics, it's very hard to find. But for whatever reason, it's present at the session level. I'm not entirely sure why.
Eric, sorry, can I ask a clarification question? This is Ali.
Sure.
So what are the rats doing during this 20 seconds? Have you looked at behavior? Are they hanging out by the food magazine, for example? Do they ignore the tone and only approach towards the offset of the cue?
So when I recorded behavior, I unfortunately did not do any video recordings, but so the condition, obviously the condition response I'm interested in is the port entry rates. And so when I look to see if there's any sort of ramping effect of port entries, are port entries sort of different earlier versus later during the cue period? The answer is no, actually. The timing that is sometimes present in Pavlovian procedures comes much, much later in this case. So it looks like their port entry rates are sort of uniformly distributed across the entire interval. Now, of course, that doesn't just mean that they're hanging out by the magazine, but so the true answer is I don't know if they're approaching the tone. Yeah. So yeah, clearly behavior is multifaceted. This is, this behavior procedure was established many years ago, and so I just sort of borrowed it. And these folks were only interested in this aspect of behavior, but more to explore in the future. So to summarize what I've said so far, delivering rewards during the intertrial interval attenuates the Pavlovian condition response without inducing selective satiety. Dopamine activity and release show diminished responses to cue and reward when cue-reward contingency was degraded. Individual differences in performance are predicted by the phasic and tonic dopamine signal during the cue outcome interval. And I'm now thinking maybe this is too strong of a response given some of the literature that's come out recently. But at the time, my opinion was that these findings were problematic for value-based accounts and reinforcement learning. So one thing I did not show you from the photometry experiments is how the dopamine response to reward changes as a function of whether the reward is delivered during the ITI versus at the end of a trial. Remember that during the contingency degradation procedure, one of the rewards is delivered not only at the end of a trial, but also during the ITI. So what I observed across experiments is that the dopamine response to the reward was much greater when it was delivered during the ITI versus at the end of a trial. And this is not terribly surprising. This has been shown before in other types of setups. But what I wanted to know is whether this non-contingent dopamine response was necessary for the learning that we see in this behavioral procedure. To address that question, I turned to optogenetics. I expressed an inhibitory opsin in VTA dopamine neurons bilaterally and implanted optic fibers that were then connected to green lasers. I then placed the rats through the same task, but during the contingency degradation phase, when rewards are delivered during the ITI, I go ahead and activate the green laser for 4 seconds to inhibit dopamine neuron activity. Here I'm first showing data from rats that did not express the opsin. Their behavior looks normal, so they show this normal acquisition, and then during contingency degradation, they show this diminishing behavioral port entry response to the degraded cue. Here are the data from the rats that expressed the opsin and experienced optogenetic inhibition of dopamine neurons. It may look like there's some sort of group effect, but in fact, the interaction is nowhere near significant, and the error bars are just quite wide. So what I can conclude from this is no, non-contingent dopamine does not seem to be necessary for learning in this procedure. So that experiment involved inhibiting dopamine neuron activity to check if this null result still holds when inhibiting dopamine release. I expressed inhibitory opsin VTA neurons and then implanted my fibers above the nucleus accumbens core. So here I'm inhibiting dopamine terminals in the accumbens, but otherwise it's a very similar procedure. The opsin has much different dynamics, so I had to adjust some statistics of the task. But during acquisition, this is the again entry rate over sessions during acquisition. Black is non-degraded, green is degraded. There's no Cree negative control group here. So what I did is during contingency degradation for 8 sessions, I introduced contingency degradation where I turn on the green laser during the non-contingent reward deliveries. And then after 8 sessions, I run an additional like 12 or 13 sessions, what I'm calling opto versus post-opto. And I compare behavior between the 2 phases. So what I'm summarizing that manipulation here in these bar graphs, and what this shows is that the effective contingency degradation is equal between the 2 phases. Once again, confirming this result that non-contingent dopamine does not seem to be necessary for the rats to learn this procedure, for the rats to learn these contingencies. And finally, I asked, is non-contingent dopamine activity sufficient for learning about contingency degradation? So to do that, I borrowed this task. It's not really a task. It's this behavioral setup that was introduced in the lab quite a few years ago by someone named Ben Saunders. He was a postdoc in the lab at the time. And what he did was to present a cue to some rats who had the CHR2 opsin and VTA dopamine neurons. This is an opsin that is an excitatory opsin. If you shine blue light on it, it'll excite the neurons, causing them to fire action potentials. So he showed this 7-second cue and then followed up with brief dopamine neuron stimulation. So I did pretty much the same thing. I have a 7-second compound cue. It's a tone and a panel light that stay on for 7 seconds. And then upon termination of that stimulus, there's 1 second of dopamine neuron stimulation given. And that's it. That's all that this procedure involves. So if you do this repeatedly, in sort of like a trial type structure across many sessions, this is the behavior you start to see. So I'm going to show you an example trial from this rat. So this rat is tethered here. This patch cord connects to a blue laser. This rat is expressed in the chenorodopsin and VTA dopamine neurons. And what you're going to see is some of the intertrial interval, and then you'll see the start of the trial. I can even play it once more in case it's unclear. So. Okay. So what I hope you noticed was that the panel light comes on, the rat becomes sort of aroused. It starts to rear, look around, and then it begins this rotation, this spinning motion. And notably, this rotation is something that was documented by Ben previously, and it seems to be a conditioned response. It's something that does not happen when you just stimulate dopamine neurons. happens during the cue period. It's sort of like an excitatory response that the animals show, and it always seems to be contralateral to the site of stimulation. Now, why does the animal show this specific response? I'm not entirely sure, but it's a conditioned response that I can then use to ask my question about contingency degradation. So what I did was I implemented this protocol for something like 12 sessions, I think. That's the acquisition phase. And then I split the rats up into these two different groups. One I'm calling maintained, one I'm calling degraded. The maintained group is maintained on the original procedure. So they get these cue stimulation pairings over and over. The degraded group also receives the same cue stimulation pairings, but in addition, during the intertrial intervals, they get free non-contingent stimulation. So it's kind of like, I'm basically trying to create a contingency degradation procedure here, but instead of using a purely Pavlovian food reward paradigm, I'm using this cue stimulation paradigm. So when I look at behavior during the final session, I'm looking both at that rearing and rotating behavior I mentioned earlier, because those are the most prominent sort of behavioral features that the rat shows during the cue. What I find is that both groups seem to show this rearing behavior, but it's the rotating behavior specifically that seems to go away during contingency degradation. So, to come back to the original question, is non-contingent dopamine activity sufficient for learning about contingency degradation? The answer seems to be yes, at least when you look at this aspect of condition responding. And as Ali mentioned earlier, there's all sorts of behavioral features you can look at. This seems to be the one that is, for whatever reason, affected most by this behavioral manipulation. Okay, so to summarize what I just told you, inhibiting neither dopamine activity nor release during non-contingent reward attenuated condition port entries. This suggests that non-contingent dopamine, at least in a canonical mesostriatal projection, is not necessary for learning contingency degradation. Stimulating dopamine activity during the ITI attenuates condition rotation. And this suggests that non-contingent dopamine is sufficient for learning contingency degradation. So not necessary, but sufficient, kind of a strange result. Can we explain that using any sort of computational model? So as I alluded to earlier, the mainstream model that links dopamine function to reinforcement learning, at least the vanilla version, cannot account for data where there's a behavioral or neural sensitivity to contingency degradation. What I've shown you are data that clearly show behavioral neural sensitivity to contingency degradation. So I simulated that model to confirm that it cannot account for any of the data I've shown you so far. But for now, I want to discuss an alternative model that can explain the findings. So as I was finishing up data collection, this paper came out. Excuse me. This paper came out from Vijay Nambudiri's lab at UCSF. It turns out that Vijay was thinking about similar issues of the role of contingency and dopamine function, and he created a computational model that specifies the role of dopamine in contingency learning. So I'll explain how the model works by omitting a lot of the math and focusing on conveying the main concepts. Suppose there's a cue reward contingency that undergoes degradation. That means that when the cue is presented, it will sometimes terminate with reward delivery, but during the ITI, much later, more reward will continue to be delivered. So let's suppose that when the cue terminates, an eligibility trace forms and then decays, representing the animal's memory of the event. If you take the mean eligibility trace of the cue whenever the reward occurs, that can give you an estimate of how frequently the cue precedes reward, which we will call the predecessor representation contingency, or PRC for short. And then you can use Bayes' rule to get the inverse of that, which is how frequently rewards follow cues, and we refer to that as the successor representation contingency. So to relate this back to the original example I showed you with my son Jude and Cheerios and tantruming, there's these two conditional probabilities we're interested in: the probability of Cheerios giving a tantrum, and the probability of tantrum giving Cheerios. one is sort of this forward-looking conditional probability, or the SRC. The other is this sort of like backward-looking conditional probability, or the PRC. And so in the context of this Pavlovian conditioning procedure I've been talking about, the relevant conditional probabilities are P reward given Q and PQ given reward. So how often does a reward follow Q versus how often does a Q precede reward? These are two different things, two different quantities. So you can compute something called the net contingency, which is just a weighted sum of these two quantities that I mentioned earlier, the SRC and the PRC, where the weight is a free parameter. So here's the sort of the insight that the sort of the whole point of the model. The dopamine response to some stimulus I is proportional to the sum of the net contingencies between stimulus I and all other meaningful causal targets, where a stimulus is a meaningful causal target if its dopamine response meets some threshold. Again, that threshold can be a free parameter. And dopamine triggers causal learning by allowing net contingency updating. So this sort of describes just the essence of the model, and there is a lot more to it that I'm omitting. But what I wanted to do was simulate the model and compare it to my empirical results. So what I'm showing you on the top row is the fiber photometry data I showed you earlier from the nucleus accumbens core. And underneath are model simulations. So we can get the model to replicate both the larger dopamine response to non-degraded versus degraded cues, the larger response to reward following non-degraded trials versus degraded trials. We can also get it to, using the same parameters, replicate the more negative dopamine response to omission following non-degraded versus degraded trials. And we can get it to replicate the strong finding that the dopamine response is larger at the time of reward when the reward is delivered during the ITI versus the trial. What about the optogenetics results that I showed? Where first I showed that when you deliver optogenetic inhibition at the time of non-contingent rewards, it does not affect the course of learning. So this is a model simulation showing the Cree positive rats versus the wild type rats. During acquisition, they show strong behavioral responding to the cue. And then during contingency degradation, just like in the empirical results, both groups show equally strong behavioral sensitivity to contingency degradation. So again, replicating what I found. And finally, looking at whether dopamine is sufficient for learning about contingency degradation, I went ahead and simulated the task I told you about, where we deliver dopamine neuron stimulation after a 7-second cue, and we once again find that the model simulations resemble the behavioral results. So all in all, this model seems to be a good description of behavior. I do want to acknowledge that since I published this paper, there have been 2 papers that have come out that have, at least one of which that has convincingly showed that you can actually, if you build your TDRL model just right, with some extra assumptions, you can actually get it to learn something about contingency indirectly. So this is an exciting area of work, I think. So I won't say that my experiments uniquely confirm this model as, you know, the be-all end-all of dopamine function, but it's certainly it's something that corroborates it, at least. So I have maybe like 10 more minutes. So I just want to spend a few minutes discussing my plans for future research. Broadly speaking, I'm interested in the intersection of reinforcement learning and basal ganglia function, as Leah said in my introduction. So when I say reinforcement learning, I mean, how do we learn from trial and error using rewards, including food rewards and addictive drugs? I actually have a whole other line of research looking at fentanyl-induced changes in learning and decision-making. And when I say basal ganglia, sorry, my cat's right here. When I say basal ganglia, I really mean a vast network of brain regions, including all parts of the striatum and their inputs and outputs. So to me, the most obvious next step is to ask whether dopamine sensitivity to contingency extends to instrumental learning. Everything I've talked about today has been about Pavlovian learning, where animals passively learn about the relationships between stimuli. And what's notable is that the computational framework I just talked about does not yet account for instrumental learning, which, as some of you probably know, is the process of learning from action feedback. This is a significant gap because one of the goals of reinforcement learning is to model how agents learn from their actions rather than just passive observation. So the first set of experiments I have lined up for the future is to rerun all the experiments I just told you about, but using an instrumental learning paradigm. And I've already started running some of these experiments, and this one is asking a very simple question, which is, does intracranial self-stimulation depend on the contingency between action and outcome? So I have rats that are pressing a single lever for high-frequency stimulation of dopamine neurons in the VTA. They learn to do this over a number of sessions. Here I only have an N of 4. Here I'm showing you the mean presses per minute over sessions. And here I'm showing it on a much finer scale. And so the animals are pressing the lever on what's called a random ratio 5 schedule. So that means that every 5 presses on average will give them stimulation. And then during the moments where they're not pressing, I deliver stimulation to dopamine neurons at random. And what's strange is that some of the rats, namely 2 of them, seem to show this behavioral effect that we normally see where lever pressing starts to go down during contingency degradation. But then a couple of other rats show the opposite. They show this facilitatory effect where now lever pressing actually increases as a result of non-contingent stimulation. And this is just showing it, the data normalized to baseline. I tried running the same sort of, or I took the same rats and I just tried replicating the result, but now under a more leaner schedule reinforcement, a random ratio of 20. And I find sort of similar things once again. So I had more animals to add to this cohort, and I had more experiments to do, but I think this is an exciting avenue of research. And I would also like to investigate whether dopamine-dependent plasticity depends on contingency. This is the illustration I showed you earlier of VTA dopamine neurons sending projections to D1 and D2 spiny neurons in the accumbens. One set of experiments that I want to pursue is to find out whether these accumbens neurons show changes in plasticity in response to behavioral manipulations of contingency, either Pavlovian or instrumental. And there's another part of this network, the basolateral amygdala, which we know encodes specific cue-reward associations and is required for the motivation to seek specific rewards. I therefore aim to focus my investigation into synaptic plasticity in the striatum on the BLA accumbens synapses. And sort of here's my pie in the sky idea. So recall the experiment I told you about earlier where they found that when you vary the timing of dopamine input into the striatum, that dopamine must be tightly contiguous with pre and postsynaptic firing in order for postsynaptic spines to show maximum growth. So I'd like to mimic this experimental protocol, but do it in vivo and then perform contingency degradation at the level of a neural circuit to figure out whether plasticity within the accumbens depends on contingency with dopamine. So this would require pairing optogenetic excitation of BLA terminals with simultaneous excitation of accumbens neurons, and following that with excitation of afferent dopamine neurons. You can think of this in like a trial-like structure where simultaneous excitation of BLA terminals and accumbens neurons is a cue, and the dopamine excitation is the outcome. And I'd repeat this over many trials, quote unquote, and then measure plasticity. And once the protocol is established, I could then conduct contingency degradation, where the only thing I change is to stimulate dopamine neurons during the quote unquote ITI. So this is what I mean by contingency degradation at the level of a neural circuit. And the results of this experiment could be interesting either way, I think, because if structural plasticity is altered by contingency degradation with dopamine, then that opens up a new line of research to figure out what the molecular mechanisms are. And it could force us to question some aspects of spike timing dependent plasticity. And of course, if it doesn't, then that would sort of bring up an interesting mismatch between behavior and neurophysiology that would need to be reconciled. Okay, so I would just like to finish by thanking first my PI and mentor, Patricia Janik, and also my postdoctoral colleague, Yifeng Cheng, Vijay and his postdoc Huizhang, who I collaborated with, who they're the ones that built the model. and then Sarah Brooke, Laia Castell, Anish Ball, and Robin Magnud for help with the experiments and the funding, of course. And I'll take any questions. Thank you.
Thank you for your talk. And since I'm host of the seminar, I guess I would just ask the first question. Since you're using like auditory cues, like what will the model change if you use other types of sensory cues?
Sorry, can you repeat? You're actually on mute, so I can only hear like your echo. Can you say that again?
I guess my question is how will the model change if you're using other types of sensory cues?
Oh, other types of sensory cues. Well, I mean, it depends, but I mean, the most obvious answer I think is that if you were to mix up the types of sensory cues where, some trials involve an auditory stimulus, others involve, I don't know, visual stimulus, you'll probably get faster learning, just because they're more differentiated. That's just usually how it works. But I don't foresee any sort of, well, actually, it's hard to predict because if you use a visual stimulus, you'll probably get the conditioned response to be sort of sucked away from the port entry here, and sort of like have more of like I forget what it's called, what Peter Holland found. But when you use visual stimuli, the rats tend to, go over and examine it, and a lot of their conditioned responding just could sort of like, be manifested in that way. But overall, I don't think it would change the findings all that much.
Thank you. Any questions from the audiences?
Hey, Eric, I'm Craig Berridge. That was a really nice talk. Nice to meet you. When you were doing your inhibition studies, the VTA inhibition, dopamine terminal field inhibition, I might have missed this. Did you confirm that inhibition functionally inhibited release in the accumbens?
Yeah, it's an excellent question because the opsin is, when I used it, was relatively new, and there had not been a lot of work done with it, but it's sort of like a check because I didn't have access to slice physiology or anything at the time. As a sort of check, what I did was after that experiment, let me just see if I can bring it up. Yeah, I have it here. So it's the lower right. So all I did was I had rats press a lever where they got food reward, and at the same time as that, they got laser delivery, so that's in green. And then another lever where they got food reward but no laser. And so this is kind of like a crude way of answering that question because it's not exactly sort of physiological evidence, but it shows what we've known from prior work, which is that when you transiently inhibit dopamine release at the time of a reward outcome, it inhibits the ability of the rat to acquire lever pressing. So this is some evidence in favor of that.
No, that's great. That's perfect. Thanks.
Question. So for some of those that you're thinking about, there's individual variation. There's individual variation, right? So have you thought about sort of whether you could look afterwards at ratios of D1, D2 in terms of responses?
Yeah, it's a good question. I will say that some of the individual variation comes about as a result of the specific sort of cue reward assignments. I mean, those are counterbalanced on the whole. All the variables that you'd want to be counterbalanced are counterbalanced. However, for whatever reason, there are some sort of cue reward combinations that sort of seem to facilitate the behavioral effects that I report. And so if I want to look for individual differences in the future, I would have to figure out a way to sort of subtract those out, which would require, I think, maybe sort of reworking the procedure, which is what I would intend to do anyway. So once that is accomplished, then I'm sort of in a position to address that question. So yeah, I mean, that's definitely something I'm interested in. I will say, though, that in this particular, in the task as it is now, the individual variation is not Actually, it depends on it depends on how you look at it. It's not huge. So a majority of the rats show the effect. So like in this example here, there's like 13 rats, 3 don't show the effect, 10 do. 4 don't show the effect, 10 do. So I mean, yeah, I mean, so the other thing is that reward port entries is actually quite a crude measure of a condition response, especially when the animals are freely moving, that it's kind of hard to use that as sort of a metric for individual variability. I think will be much better is kind of going on with Ali. What Ali was suggesting was maybe using a sort of like a like a sort of like a compound measure, like between like many different aspects of the animal's behavior, and maybe that could sort of that could increase the amount of variability that I could play with.
Go ahead.
When they're doing that turning behavior, does it look like they're being vigilant? So are the ears up, and could it be a type of vigilant behavior?
A vigilant behavior.
I mean,
So,
usually vigilant behavior, we often think about it directed to a stimulus from one location, but I don't know.
Well, I will say that I do, I mean, so the ears being up, I mean, so is that something that happens with rats? Like their ears will go up when they're vigilant? I don't know enough about that. But I will say that I did.
California mice do that. I actually have no idea whether rats do that.
Okay,
I would
well,
assume it's alertness too. For example, if there was a danger in the environment, it would be alertness towards it. I mean, this is sort of more searching almost, but
yeah, I mean, so I
figure out what it means.
I did track the ears. I didn't see any sort of
Okay.
changes in the ears, although, you know, this sort of angle, this camera angle may not be sensitive enough for that. But in terms of what the behavior means, I mean, that's an excellent question. And the postdoc who I mentioned earlier, Ben, who sort of invented this procedure and has been following up on it, I don't know if he necessarily knows what it quote unquote means, but it's an excellent question. And I kind of get that question a lot, so I think it deserves some investigation. So I'm sorry for my
Is
answers
it
on
searching, like when they find something, what causes them to stop doing it?
Well, that's a good question. I mean, so the stimulus is on for 7 seconds, which is kind of a short amount of time, at least for these types of experiments. So what I generally see is that the rotation, the probability of rotating increases along that interval. So if we made the interval like really long, I would just assume that the rotating would still come at the end. It would be like time invariant. So what causes them to stop is, I think, that the termination of the cue, either that or the dopamine stimulation itself. But I think it's just the termination of the cue. So I don't think it's like goal-directed, purposeful behavior. It's just some sort of
Searching.
conditioned reflex. I mean, in the true sense of the word.
So I have a naive question to follow up on that. I remember this old literature when unilateral, I think it was depletion of dopamine would induce this stereotyped rotation. Is this any way related to that phenomenon? Do you know what I'm talking about?
Yep, yeah. So you are correct that there's a large literature showing that when you lesion dopamine neurons, or if you lesion dopamine in the striatum, you'll get these sort of like contralateral effects where the animal will turn in the contralateral direction relative to where the lesion occurred. So you could possibly argue that these rats have lesions because I've been stimulating them like crazy, but we don't see this rotation at baseline. So if you measure 7 seconds prior to the trial, the sort of rotating behavior is not there. It's only conditional upon the cue. But in addition, we do know that dopamine is lateralized. So if you were to record dopamine release during like a task where the animal has to choose between different sides of a chamber, for example, dopamine neurons will show this lateralized signal. But why it is that the animal turns specifically, I mean, and not some other lateralized behavior, again, I'm not entirely sure, but yeah, laterality and dopamine go way back.
That makes a lot more sense, because it doesn't actually look that natural. So I kind of like those explanations a little better.
Yeah.
But
So I was a question about it. This rotation is not about the lesion, it's about the imbalance of the receptors.
That's
Could be a cue about that probably when there are degradation, the expressing of the receptors are gone. So then you don't have imbalance. But when you don't have degradation, then the receptors are overexpressed for the continuous releasing of dopamine. And then when these turn on, to create an imbalance. So the rotation behavior of the Parkinsonian unilateral mice was not created for deletion. It's creating for the imbalance because one side has not receptors and the other side has receptors. So then you create these motor loops. Could be a good cue about like plasticity, in my opinion.
I'm afraid I didn't hear a lot of that. I'm sorry.
Sorry. Is it this thing that Stephen is? Okay. Sorry if you.
So you can hear me?
Yes.
Okay. So I was saying that they mentioned that this rotation is created for lesions. But actually it's not for the lesion. It's about that you are creating an imbalance between the hemispheres and one hemisphere has receptors of dopamine. So it's possible that your rotation is caused because in one condition, you have more receptors of dopamine than the other condition. So this is why you saw rotations.
Okay, that's interesting. I just one thing that would need explaining, I think, is how this. So where in this experimental setup, I'm doing unilateral stimulation in the midbrain.
But if this dopamine increasing will be in accumbens.
Right.
Yes. So then, but one side, you are turning on only one side.
Right. I think, did you say something about the number of receptors possibly being
Yes.
different between hemispheres? Do you think that this optical manipulation could somehow be changing the number of
I
receptors
think
between?
it's your behavior paradigm. It's like in one is degradation and the other one is maintained, right?
Yeah.
But you don't see these rotations. It's different.
Right.
So it could be a cue of the plasticity.
Interesting. Oh, that's an interesting idea. Yeah, I guess so. Would it be sufficient to show that? I guess like in order to support that explanation, one would have to show that, giving this number of dopamine neuron stimulations optically sort of changes the balance and the number of receptors between
Yes,
hemispheres and the
because
striatum.
it's
Yeah,
classic for imbalance in Parkinson.
right.
Yes.
Yeah, thank you for clarifying that. I didn't realize because I thought that the literature was on 6-hydroxydopamine lesions in the striatum.
No, it's about that you have more receptors in one side and not in the other.
Interesting. Oh, well, I just learned something new.
Thank you.
Okay, I have one question. So like I'm interested in not only learning effects of dopamine, but like other behavioral effects, such as motivation, right? And then one of the graphs you showed, I think, is alluding to that. I mean, the one you showed that there's a positive correlation between dopamine magnitude and number of head entries, right? So do you think that one, right? Exactly. So do you think, I'll get to the differences between phasic and tonic. Do you think that one supports the graph on the left supports the motivational role, that if you get a big dopamine boost, you're more likely to approach and poke?
Yeah. I'm afraid that this sort of analysis does not cleanly distinguish between, questions of motivation versus learning, unfortunately, because I think, because you could easily, conceive of these graphs as showing what you were just saying, which is that like the motivation to respond is supported by, larger phasic versus tonic signals, and And I mean, I assume that the learning has already occurred by this point. These are data taken from the final session of contingency degradation, where the behavioral effect is maximum. They seem to have learned the contingencies by this point. Now that doesn't, of course, there's within session learning that's always sort of occurring because the rewards are probabilistic. Nevertheless, however, because the learning is more or less sort of at its termination at this point, I think this could actually be sort of an argument in favor of sort of a motivational.
Favor that. Yeah, and I think if you go to pop culture, you can say that if you get a dopamine boost, you're more likely to get up and move, right?
Yes.
And then you can take it to some podcasters maybe there. Well, no. Okay, thank you. Yeah.
But you're gonna ask.
Oh, differences between phasic and tonic, I think also are very interesting. That also is correlated, right? So it's not just the initial bump. And I don't know if you've looked at the correlation between phasic and tonic.
Yeah. So the reason why both these plots show positive correlations is because phasing and tonic signals are themselves positively correlated with each other, but within rats, which I mean, I know this is Ali, I know this is sort of like, your bread and butter. I don't know if that's surprising to you.
Kind of. I mean, the trace you were showing, which showed multiple prolonged release events, right? It was not just elevated levels.
Right.
So I think that's that one, right? So I'm wondering if when the animals get a big phasic response, it would induce more kind of behavior that would cause dopamine release, right? That is not just the tonic levels are going up, that you have more dopamine related events.
I see.
I don't know.
Yeah,
They start scratching, they start doing things, right? I mean, they start moving around and maybe poking inside the magazine and doing all these things that would cause individual release events. And then when you sum it up, it looks like higher tonic levels.
Yeah.
Maybe.
All the more reason to track other aspects of the animal's behavior.
Yeah, fair. Mm-hmm. All right. Thank you. Yeah.
Yeah, thank you.
Any more questions right now? I guess. Okay. I guess we're at time right now. If you have any more questions, please email Eric directly, or I can forward your email to him. Thank you all for participating this week, and thank you, Eric.
Thank you.
Thank you very much. Appreciate your time.
Yeah, thank you for hosting. All right, goodbye.